Hi everyone! For Chapter 3, we will talk about the four main categories of biological macromolecules, what they're made up of and how they are made. There will be a lot more terms you have to know and remember from this chapter, so be sure to take good notes, make flashcards to help you be very clear which monomer goes with which polymer, what type of bonds are made in which macromolecules, what are those macromolecules called, what are the specific examples, how you can recognize them by images or illustration will be what you need to focus on for this particular chapter, okay, on the exam. All right, so let's get started. So the four major classes of biological macromolecules we will discuss in this chapter are carbohydrates, lipids, proteins, and nucleic acids. All of them are essential, okay, meaning that they are necessary for survival of life. Hence, that's why we call them essential. Now, since they are macromolecules, which means they tend to run pretty large, I'm talking about in terms of molecular weight and in terms of size, how many atoms they're made up of, etc. So they are made from smaller components or subunits called monomers, or what we call single subunit. Okay, so mono for single, that's why it's called monomers. And when multiples of these monomers come together and form covalent bonds with each other, they form polymers. Okay, and a lot of times when monomers come together to form polymers, they have to be bonded by covalent bonds. Okay, now you understand why in chapter 2 we have to went over all those different bonds that can be formed between atoms, because they will become applicable in this particular chapter for forming these polymers. So poly, as you can already imagine, poly means many, which means lots of stuff coming together. Those stuff is molecules, subunits, etc. So those are what macromolecules are. Okay, so macromolecule stands for those large polymers that are made. So the process of making polymers from monomers by forming these covalent bonds is called dehydration synthesis. Okay, dehydration synthesis. So for example, one of these hexagons right here in this picture, those hexagon shaped molecule represents a single glucose molecule. Okay, so this right here is a glucose. And two glucose molecules, both of these are the same thing. When two glucose molecules come together, they can form a covalent bond with each other to produce a disaccharide called maltose. Okay, so this is a disaccharide called maltose. Okay, not that the whole name is called disaccharide maltose. I'll explain when we get to the carbohydrate section, I will discuss what we mean by monosaccharide versus disaccharide versus polysaccharide. But for now, just go with the flow. Okay, so the 2-glucose is what is able to make a molecule of maltose and water. Water is a byproduct right here. Okay, so this is water right here, water molecule. This here is the maltose. Okay, and you see that this is the covalent bond that is formed between the two molecules of glucose. Okay, so there was no bond between them here, and then there's this bond right here joined by an oxygen in the middle of them. And we'll talk about what those specific bonds are called depending what type of macromolecule it is. All right, so whenever we have a dehydration synthesis reaction, water will always be a byproduct at the end. Okay, so that's one clue you have as to whether this is a dehydration synthesis reaction. And synthesis should give you the idea that you're forming something. So you're forming a bond. And the fact that it's called dehydration, okay, dehydration, just like how you dehydrate when you run out of water, because you're removing water. Okay, so that's why it's called dehydration. dehydration reaction. You're removing a molecule of water from the reaction to form a new bond between two different molecules right here. Okay? All right, and the water molecules, where it come from is you have this hydroxyl group, OH, and here's the other OH group, okay? Here's the other OH group, okay? So this hydrogen from this glucose molecule and this oxygen hydrogen, which we call hydroxyl group, from this glucose can come together, remove these two things to become water, H2O, and this oxygen here will then become bound to this carbon right here instead. Okay, that's how you form this bond between the two glucose molecules. Now, what about when we need to break a bond? Okay, we need to form a bond, but in order to produce energy, we also need to form a bond. Oh, and before that, before talking about breaking a bond, whenever we're forming a bond, it requires energy. Okay, require energy, meaning that it must utilize energy coming from another source. Okay, and hence we'll talk about energy flow in a system, in a biological system, when we get to chapter 6, because we need to know where this energy comes from. Okay, and hence this is why we need to eat. This is why we need to gain energy, or in form of ATP when we're digesting those food. And why we need those energy is there are some chemical reactions in our body that will require energy. Okay, and usually those that are forming bonds require energy. Okay, so make sure you remember this concept. Dehydration synthesis is forming bonds and it requires energy. Okay, so now what about when we break a bond? What happens there? So whenever we break a covalent bond between a polymer and split them back into monomers, this process is called hydrolysis reaction. This time, instead of removing water, okay, that's why it's not called dehydration, we are adding water, okay, and that's why we call it a hydrolysis reaction. Hydro, remember, easy way to remember that you're adding a molecule of water is, we all know hydro, as we've discussed last chapter, means water. So it means we're adding water to the reaction. Okay, one is dehydrate, removal of water to form a bond. Hydrolysis reaction, you're adding water to break a bond. Okay, so again, that should be something you write down as on your notes or as a flashcard or something, so you don't mix up the difference between dehydration synthesis reactions and hydrolysis reaction, and when is water molecule added or is it formed as a byproduct. Okay, so what happens with water is when you add a molecule of water right here, okay, it's going to do the opposite of what we did in dehydration synthesis. Dehydration, we remove H and OH from the two glucose molecules to form the bond. Now we're going to add water so that one of those H's are going to come with this glucose, and then OH group will be added to this glucose, okay? And hence that's why you end up with a extra hydroxyl group in this glucose and this extra hydrogen in that glucose. Okay, so this was the original maltose we have, where the two glucose come together, and then now we're breaking them back to what it was before. Okay, when we are trying to, I'm trying to get my pointer here, when we're trying to form bonds. Okay, so we're just getting that right back here. All right. And with the opposite of synthesis of polymers, when you break a bond, break polymers, you are breaking bonds, and it always release energy. Okay, so when you're forming a bond, it requires energy, but when you're breaking a bond, it releases the energy that is stored within. We kind of talked about it last chapter when we talked about why hydrocarbons are a lot of times used as fuel source due to the high amount of energy they can store in all those covalent bonds. Okay, so that's the same thing here. We're breaking covalent bonds and hence we are releasing energy from the reaction. Okay, and we will, like I said, we will come back to these ideas of energy when we get to chapter 6 on energy metabolism. All right, so let's get started with our first category of biomacromolecules, our carbohydrates. Okay. So there are 3 subtypes of carbohydrates, like I was talking about. You have monosaccharides, disaccharides, and polysaccharides. And they're kind of easy to remember because we know what mono means, we know what di means, and we know what poly means. Okay, mono means single. Di means 2 or double, if you like to think of it that way. And poly means multiple. Okay. And I was saying, so when it's monosaccharides, it means 1. Okay. Mono mean 1. Saccharides stands for something that has to do with sugar. Okay. So carbohydrate of some sort. So saccharide is the root word for carbohydrate, and the prefixes tells you how many carbohydrate or sugar molecules are there in this particular macromolecule. Okay, so monosaccharides is just 1 sugar molecule, okay, which are usually, really the simple sugars. And a classic example of a simple sugar is glucose. Okay, and for carbohydrate, they usually function as a storage of energy. Okay, so that's the main overall theme function of carbohydrates. They are for storing energy because they have, remember we talked about hydrocarbons, they have a lot of C's and H's and lots of little bonds there for us to break to release energy. Okay, all right, so the second, first of all, monosaccharides can be further characterized by 2 schemes, okay? Or, sort of set it as a nomenclature kind of thing. We can name it 2 ways. The first scheme, or, first naming scheme, has to do with where the C double bond O, or what we call the carbonyl group is, okay? So carbonyl group, that is the C double bond O, C double bond O. That's called, remember we talked about application of functional group last chapter. We ended with that, and I said we're going to talk a lot more about these functional groups in chapter 3. So we learned that OH is called hydroxyl. Okay, so OH group is called hydroxyl group. So C double bond O is called a carbonyl group. Okay, so carbonyl group. So depending where the carbonyl group is, it is either called ketose or aldose. So if the carbonyl group is at the middle of the molecule, where you have a carbon and a carbon attached to that carbon of the carbonyl group, we call that a ketose. Okay, so there's a carbon attached, there's a carbon attached. Both sides are carbons attached to the carbonyl group. Okay, so we call that ketose. You can kind of remember it kind of looking like a key there. Okay, looking like a key. And then, the zigzag a key. Okay, and then for carbonyl group that is at the end of the molecule, either this end or this end, it doesn't matter, but one of them, because it's at the end, it shouldn't be attached to a carbon. One of them, the carbon of the carbonyl group right here, will be attached to a hydrogen instead of a carbon. Okay, so you have a hydrogen attached to one of it. The other one is a carbon, of course. And not both sides are, sort of sandwiched between 2 carbons. It has a hydrogen attached to the carbon of the carbonyl group. We call that an aldose. Okay. Okay, you don't have to know ketone versus aldehyde group, but you'll learn that in chemistry. But for here, we're interested in how we can categorize the glucose or your carbohydrates by where the carbonyl group is located. Okay, so for glyceraldehyde is the name of this particular sugar right here, or carbohydrate molecule, because the carbonyl group is at the end of the molecule where you have one hydrogen attached and the other side is a carbon. We call that an aldose. In this dihydroxyacetone carbohydrate molecule here, you have the carbonyl group in the middle where you're flanked by two carbons, so we call that a ketose. The second scheme of nomenclature of carbohydrate or monosaccharide has to do with how many number of carbons does the monosaccharide contain. So this one is pretty easy. If you know your geometric shape and you know how to count how many C's are there in the molecules, it should not be a problem on the exam. Okay, so when we're talking about how we label monosaccharides by number of carbons, if it has 3 carbons, we call it trioses. Okay, oses here stands for the fact that it's a monosaccharide and is part of the sugar group. That's why we have glucose, okay, ose meaning something to do with sugar. And then 5 carbons, when you have a monosaccharide with 5 carbons, just like geometric shape with 5 corners, you call it a pentagon. So for sugar is a pentoses, okay? And then when a sugar molecule or monosaccharide has 6 carbons, you probably guessed it, hexagon, right? For a geometric shape when there are 6 corners. So, now you have 6 carbon. We call it hexoses, okay? Hexoses. So this is an example of a triose because you have 1 carbon, 2 carbon, 3 carbons. Okay, that's all the number of carbons there is in the glyceraldehyde molecule. So it's considered as a triose. Ribose, okay, is ribose right here. Has 1, 2, 3, 4, 5. Okay, 5 carbons, so we call it a pentose molecule. And lastly, when we look at a molecule of glucose, you have 1, 2, 3, 4, 5, 6 carbons. So it's called a hexose. Okay, so make sure you remember these 2 schemes of naming monosaccharides. Okay, other than their actual specific chemical name, we can also identify them as a ketose, aldose, or by the number of carbons as trioses, pentoses, or hexoses. All right, so 3 specific examples of monosaccharides we will talk about here are glucose, galactose, and fructose. All 3 of these are structural isomers of each other. Remember, we talked about different types of isomers in the last chapter. We talked about structural isomer, geometric isomer, and enantiomer. So these 3 particular carbohydrate or monosaccharides here are specifically structural isomers of each other. Okay, remember, structural isomers are when they have the same chemical formula, but the structural bonding of some functional groups are different between the 3 molecules. So all 3 of these have the same chemical formula of C6H12O6, okay, meaning 6 carbons. They all have 12 hydrogens and 6 oxygens. But how the different functional groups are arranged within the molecules are different. That's why we can't call them by different names, okay? And they're not the same molecule. Now, can you practice and tell me whether each of these that we're seeing in this image here, by each one of these, which ones are aldose and which one is a ketose? Take a moment. And then I will go over it together. So look at glucose. Where is the carbonyl group? Carbonyl group is a C double bond O. That's right here. Is it attached to 2 carbons or does it have a hydrogen attached to the carbon? It has a hydrogen attached to the carbon. So glucose is an aldose. How about galactose? Same thing. We have a carbonyl group right here, and it has a hydrogen attached. So galactose is also considered as an aldose. Fructose, however, here is the carbonyl group and is attached to one carbon, two carbon. Okay, so it's attached to carbons on both sides of the carbonyl group. So it is a ketose. Okay, and hence that's why you have glucose and galactose as aldose and then fructose as ketoses. Okay. Now you might ask, why is glucose linear all of a sudden in this representation right here? For example, in this one, why is glucose linear when we just saw it, a few slides ago as a ring or hexagonal structure? Well, that's because there exists an equilibrium between some 5 or 6 carbon monosaccharides where they can sort of interchange between linear forms versus carbon ring forms. Okay, so whether this representation of glucose or this representation of glucose right here, they're still the same thing, because technically speaking, in a chemical, from a chemical standpoint, it is an equilibrium where you have, there should exist both forms of the molecule. Okay, but we're not going to dive too deep into that realm of chemistry. Okay. All right, now if I went on to ask these 3 molecules right here, if we want to name all 3 of them by the number of carbons, what will we call these 3 molecules? Is it triose, pentose, or hexose? Right, they're all going to be hexoses, okay, because all 3 of them have 6 carbons in there, okay? All right. So when 2 monosaccharides come together to form a polymer, we call that a disaccharide. Di, remember, that means 2 or double the saccharides, which is the sugar molecules. Okay, so again, they are formed by dehydration reaction. Okay, dehydration reaction is when we're forming bonds, and it happens by removal of a water molecule. Okay, which means water will be a byproduct in this particular reaction. Okay, so disaccharides are formed by 2 monosaccharides coming together, and there are 3 common types of disaccharides. You have to know what they are and which monosaccharides produces them. Okay, this is something you will have to memorize, okay, and remember. So for lactose, okay, as all of you probably know, lactose, lactate, okay, kind of sounds like something that has to do with milk. Okay, so lactose is a particular disaccharide that is naturally in milk products. Okay, so, or dairy products. So lactose come from a dehydration synthesis reaction between glucose and galactose. Okay, so make sure you remember that glucose and galactose come together to form lactose. And it's not too hard to remember because galactose itself has the name lactose in it. Okay, so if you were to see, let's say, a multiple choice question that says galactose, you're like, oh, okay, I know. Okay, galactose is for making lactose. It has its name in it. Very straightforward, easy to remember that one. And then maltose, as we have talked about in the beginning as the first example of dehydration synthesis, is formed by 2 glucose molecule, and maltose is usually what's in our grain sugar. Okay, when we have those grain sugar molecules, grain sugar products. And then in sucrose, okay, sucrose right here is when we have a combination of 1 glucose molecule with a fructose molecule. Okay, so these 2 monosaccharides here come together to form sucrose. Fructose, glucose makes sucrose, and sucrose is a common macro or disaccharide that is in table sugar, which is the sugar that you put into your coffee this morning, for example. Okay, and this shows you how these molecules look like. This is maltose that we saw in the beginning as the first example. This is lactose right here. And then you have sucrose. And you see they all have this oxygen in the middle as a way to form a bond or covalent bond between the two monosaccharides. Now, make sure, remember I mentioned that you will have to know the individual bond names for each type of biomacromolecules. So for sugars, the covalent bond that is formed between two monosaccharides is called glycosidic bond. Okay, and it's, easy way to remember that is the word begins with glyco, okay? And glyco, as we will talk about in a bit, it has to do, do with or related to glycogen, which is a type of carbohydrate polymer. Okay, so just remember glyco sounds like some form of a sugar, so it's got to be the glycosidic bond that sits between two monosaccharides or multiple monosaccharides to give you disaccharide or polysaccharide. Okay, so that's what I mean by there are a lot of new terms in this chapter that seems to all jumbled with each other, and every semester students get confused with which bond names go with which macromolecule or which monomer goes with which macromolecule. So that's why it is very helpful for you to start forming and drawing these flashcards, so you have a group where it's a bunch of information about carbohydrate, and then a group of flashcard for lipids, and a group of flashcard for proteins, and then your nucleic acids. Okay, so that way you don't get confused between all these terms that's thrown at you in this chapter. Okay. All right, so the glycosidic bond is the covalent bond between carbohydrate and another molecule. So this right here is the glycosidic bond like I was talking about. So that oxygen in the middle and the new covalent bond that is formed between this monosaccharide and this monosaccharide, which is glucose and fructose, to make a sucrose molecule is by forming this glycosidic bond right there. Okay, and whether it is a maltose, lactose, or sucrose, the bond that forms between two monosaccharide is always called glycosidic bond. All right. When more than two monosaccharide come together in covalent bonds, forming a long chain of carbohydrate, we call it a polysaccharide. Remember I said poly for many, saccharides for sugar. So many sugars coming together. So some can be one straight long chain in the unbranched conformation. Some can be branched like a tree. Okay, because how chemicals come together, it doesn't have to be always like this. Okay, they can be long chain, of course, but sometimes we can be like this. Okay, and then start branching off some more as well. Okay, so this is what we mean by branch versus unbranched, depending how the monosaccharides are coming together and which particular polysaccharides are they. All right, so common examples of polysaccharides that we may see and eat even are your starch molecule, glycogen molecules, cellulose, and chitin. We will go through what each of these are next. So there can be two specific types of starch. We'll start with starch first. So starch from plants is when they store sugar molecules in their cells, because plant is known to make sugar as well, okay, to supply themselves with energy, and also we humans as consumers can consume these plant produce fruits in form of a fruit or whatever it is with all those starches, and we get energy from them too. So that's why plants will make their own sugar molecules, and they will have a way to store them. And usually they can form either unbranched or branch version. The unbranched version, like this, remember I said unbranched should be one linear molecule structure. So this one linear molecule structure in the unbranched version is called amylose. Okay. Amylose, we're talking about sugar that is produced specifically by plant cells, okay, by plants. Not by humans, okay? Humans don't make amylose. And then you have Those that are branched, okay, is called like this, branch. Remember, I talked about how branch can be like this. So this is what we can see as a branched version of starch molecules from plants, and we call those amylopectin. Okay, so amylose for unbranched, amylopectin for branch versions of the polysaccharide starch that is specifically made in plants. You will observe where potatoes store their sugars inside their cells in lab. When we get to the particular lab called cells, rice is another example where they're abundant in amylose and amylopectin. Okay, so those are just few examples of plants that we ingest for their particular sugar resource. So the other type of polysaccharide is called glycogen. Okay, these are specific examples of polysaccharide. So glycogen, which are storage form of glucose, and let me erase that so that's not in the way. All right, so you have glycogen, which is storage form of glucose in humans and animals, and they are usually always highly branched in particularly the liver cells and muscle cells. Okay, so this is how, for example, a glycogen can look like in the human body, for example, very highly branched all over the place, and usually for us, they're always stored within the liver and also within muscles. Okay, so that's our fast resort of glucose for energy. Okay, lots of glucose can combine together to form glycogen molecules for quick resource of energy. Once we run out of glucose and glycogen, then our body then will turn to fat for energy, which we'll talk about as our next macromolecule. All right. And hence, make sure you remember how these pictures look like. I might ask for and show you these images of the different macromolecules and ask for what is this called, or what is this called, what is this called, etc. Okay, are these in plants or are these in humans? If they're in humans, where specifically can we find glycogen, for example? So make sure you do, highlight all of these important points I've talked about in this particular lecture. All right, so a third specific type of polysaccharide is called cellulose, and those come from plant cell walls. This one is pretty straightforward. Cellulose for plant cell walls, and if we zoom into the molecular level of plant cell walls like this, so for example, this is a plant, and within the leaf, if we look at their individual cells, and then we zoom in to these particular green borders we're seeing around the cell, this is what the cell wall looks like. And you will see that they have many cellulose fibers that overlap with each other to form these fibers, okay, all these little fibers here that we eat from our vegetables, like celery, for example, lots of fibers there. So the cellulose is very important in plant cell walls to provide them with structural support. Okay, not only we humans, after eating plants, all those fibers from the cellulose there is helpful for our digestive purposes, as those fibers are moving and sweeping past our intestine. But for the plant itself, they serve as that very essential component of structural support. Because in plants, they don't have skeleton and bones like us to uphold their body against the pull of gravity, but yet they grow pretty tall. We have trees, and we have bushes, bushes, or flowers. Sunflower, for example, those go up from the tree, grow up from the ground. So those without skeleton, how can they support themselves? Is by formation of these cell walls in their plant cells. Okay, so what keeps them standing upright and not curve all over the place is due to cellulose providing that strength in the overall structure of the cells. And lastly, chitin is the last specific example of a polysaccharide, and this is a polysaccharide that contains nitrogen in it and is is usually used in arthropods. Okay, you'll learn next semester, if you're taking 1407, that there's a group of organisms called arthropods, and usually those includes insects and crustaceans. Okay, insects, you know what those are. Crustaceans are usually shelled organisms like crabs, shrimp, etc. So those usually have a very hard surface as their exoskeleton that protect them from, predators or whatever it is that they're living in their environment, and can avoid predators being eaten by other animals, etc. Okay, so the big component of that exoskeleton is chitin. Okay, the polysaccharide chitin right here, that's within the shells of this beetle or within the shell of this crab. Okay. All right, so make sure you remember cellulose for plant cell walls, and chitin for being part of exoskeleton in crustaceans or insects, or, in general, we call arthropods. Okay. All right, so now let's move on to the next main biomacromolecule, lipids. Now, for lipids, these can be a little bit more confusing because lipid categories can look very different from each other. As we have mentioned in the last chapter, lipid molecules are usually hydrocarbons that tend to be nonpolar or hydrophobic compounds. Okay, nonpolar, hydrophobic. So they tend to repel water molecules. Remember my little picture from last chapter about the oil versus the vinegar, and how the vinegar kind of formed little dark circles in olive oil because the oil and water don't mix. So same with lipids. Okay, they are just, nonpolar and hydrophobic things. Just as we said, hydrocarbons usually serve as fuel for our cars or lighters. There are also lipids in our body that serve as long-term storage of energy in similar way. Notice the keyword there, long-term storage. So this is different from glycogen. Okay, glycogen from carbohydrate as a polysaccharide stored in the liver and muscle are our fast resource of energy. They get burned, they will get broken down, and only when we deplete our body of mostly everything else, then we come to our lipids. Okay, which is why they say it's hard to burn your fat. You gotta at least be running on the treadmill for 15 minutes or more before you start burning fat reserves, because that is our long-term storage of energy, and it's sort of like a second resource. Okay, because our first resource is always glucose and then glycogen. And this is something you will learn when you get to anatomy and physiology if you're going down that route. Okay, so there are lipids that are for structural purposes as well. Okay, so not only do they serve as storage of energy, there will be ones that are for structural purposes that we'll talk about in a bit. Okay, so hence we will discuss 3 different subtypes of lipids: fats, phospholipids, and steroids. And these 3 subtypes are quite different from each other. So make sure you're very clear which is which, how they look like, and what are some key features you can identify them as. So let's start with fats. Fats are usually made up of 2 different components. The monomers for carbohydrate is not glucose molecule, but glycerol molecule. For monomers for carbohydrate is glucose molecules, while the monomers for fats, are 1 glycerol molecule and 3 fatty acid chains. So they together, that 1 glycerol molecule and 3 fatty acid chain, they form a larger polymer called triglycerides. Okay, that's why it's called triglyceride. Tri meaning that you have 3 different molecules of 5 fatty acids being in the molecule. Okay, and they form your fatty acids. They form your fats. Okay, so now here is that glycerol group. Okay, glycerol group, and then you have your fatty acid chain, one of them anyway. And fatty acid chains are really just long chains of hydrocarbon molecules with a carboxyl group at one end and a hydroxyl group, because this hydroxyl group has to react with these hydrogen molecules right here within the glycerol to form a covalent bond. Okay, so you have your triglyceride right here, where you have one fatty acid chain, 2 and 3 fatty acid chain, and this after dehydration synthesis that come from the hydroxyl groups of the fatty acids and the hydrogens of the glycerol, you form this same as that what we talked about in glycosidic bond. You have that oxygen molecule serving as a covalent bond between the glycerol and the fatty acid. And instead of calling this glycosidic bond, we call this the ester bond. Okay, I know I didn't write this down as a bold underlying word, but because in lab you will learn about the different bonds from 4 different macromolecules, you will have to know that bond name anyway, so might as well write it down here and remember that. Okay, so ester bond is what we call the bond that formed between the glycerol molecule and the fatty acid chain. All right, so there are 3 different specific types of fatty acid chains. There are saturated versus unsaturated. So saturated fatty acid is when there are only single bonds between carbons. So if you look at all these carbons here within this whole chain of fatty acid, they're all bonded together by carbon single bonds. Okay, now we're not talking about the carbonyl group here. We're talking about these carbons here along the whole chain, the hydrocarbon itself. Okay, so they're all single bonds, no double bonds, and hence they are saturated with hydrogen molecules all over. That's why there's all these H's all over the place. That's why it's called saturated fatty acid, because they are saturated with hydrogen atoms. And because they are formed as linear conformation in 3-dimensionally speaking, they tend to stack on top of each other in an easy format, and that's why usually saturated fatty acid can become solid at room temperature. Okay, and hence that's why you sometimes see some fat starting to look looking like goo almost. If you've ever cooked bacon on a pan, when you cook bacon on a pan, all these grease comes out of the bacon itself, but once the pan cools down, you see all these white stuff in your pan if you didn't wash it right away, and they're kind of gunky and stuff. So that's that saturated fatty acid being formed, starting to form more solidifying or solid structure when it's at room temperature. Okay, and those is what can contribute to plaque formation in arteries. So that's why they say, we can ingest saturated fatty acid, but make sure you ingest them in moderation, because if you eat too much saturated fatty acid, which is usually common in red meat, then that can lead to a bigger risk for cardiovascular disease due to those plaque, then, adding to the plaque formation within the arteries or your blood vessels. Okay. Unsaturated fatty acid is when there exists a double bond between 2 carbons along the molecule. So here is the whole molecule of hydrocarbon chain right here. Well, there's a single bond all over the place on this side of the molecule and this side of the molecule. All of a sudden you see the C double bond C right here. Okay, C double bond C, that's that double bond we're talking about. When it has at least one of these double bonds, we call it an unsaturated fat. Those unsaturated fat with only one or single double bond is called monounsaturated fat. So this particular example here is a monounsaturated fat. Those with multiple double bonds are called polyunsaturated fat. And we'll talk about specific examples of that in the next slide. So we have already discussed in the last chapter how this particular molecule this one right here, can have geometric isomers in the cis conformation and the trans conformation. The cis conformation was when the hydrogen sticks out in the same side of the molecule. You see these hydrogens right here, they're on the same side of the C double bond C. Okay, they're not across from each other, they're on the same side, which creates this, sort of this kink or bend in the molecule. So the hydrogen is on one side, and these two bulky groups of hydrocarbons are also on the same side, because we need to make room for these bigger bulky group. That's why we create, this molecule ends up with a little bend like this that creates a kink, okay, in the molecule that allows it to be liquid even at room temperature. Imagine, I kind of talked about this last chapter, how I said linear stuff, they can stack on top of each other nicely to form a big block of solid, whereas when you have kink molecules like this, yes, they can technically stack like that, but because of that kink in there, it's less likely that they will stack on top of each other like this, because that angle that's formed by the kink has to stack on that exact same spot, versus here they just need to align with each other like this to start stacking on each other. So whenever you have a kink in the three-dimensional shape, it is harder for them to stack on top of each other, and that is why cis unsaturated fat is something that is, technically speaking, pretty good for our body. Okay, there's unsaturated fat. They are liquid at room temperature, so they're not there to form plaques. They're known to help even lower blood cholesterol level, because of their unique three-dimensional shape, with that kink inside the molecule, okay, or bend within the molecule this way. Okay, so saturated fat, it's okay, but when you eat too much, they can start to form plaques like this by stacking on top of each other. When you have a kink in unsaturated fat, that can help lower blood cholesterol, because they are less likely to stack on top of each other. And transconformation is the other geometric isomer we talked about in the last chapter, where the hydrogens are on opposite sides of the carbon double bonds. Okay, so you have your carbon-carbon double bond right there in the hydrocarbon chain, and then you have the hydrogen one on this side, and then the other hydrogen on the other side. And because now you have this big bulky hydrocarbon group, and this hydrocarbon group on opposite side of the carbon's double bond, on opposite sides of the carbon double bond C, you don't need to have that bend, because we're not sort of three-dimensionally making room for the bulky group. Okay, so that's why trans fats, okay, in trans configuration, they tend to be very similar to saturated fat in a way that they can stack on top of each other like that, okay, in forming plaques. Okay, so that's why trans fat is not good for our health as well. And let me erase that. Okay, so let's see. So we actually don't make trans fat naturally. That's something for you to know. It is made by human techniques, where food industry artificially, notice the word artificially, that means we don't naturally make this. They artificially hydrogenate oils to make them semi-solid, because remember we talked about unsaturated fat is usually liquid at room temperature, and saturated fat is usually solid at room temperature. Well, turning that into trans fat is what makes them semi-solid, where we're turning cis fat into trans fat. And the whole, in the beginning, why they were doing this is to make shelf life of, for example, margarine, and shortening a lot longer in stores so that they don't have to throw oils that is not sold in down the trash. Okay, if you have a longer shelf life, they can earn more money from that. Okay, which is the whole reason why they started the whole industry of trans fat in margarine and shortening. However, they are not very good for our bodies. Okay, trans fat will lead to higher LDLs. Okay, LDL is our bad cholesterol. I'm not going to go into the difference between HDL and LDL here. You'll learn about that in an anatomy. But LDL is our bad cholesterol, and trans fat has been shown to lead to higher amounts of bad cholesterol and also increase the risk of plaque formation in arteries, which means increased risk of cardiovascular disease. Okay, dealing with your heart. So just a change in geometric shape can completely reverse their effects in our body when comparing between cis and trans fat, because technically speaking they are the exact same chemical formula, but we're just changing their geometric shape into their different geometric isomers, cis versus trans. Cis conformation can help prevent cardiovascular diseases, while trans fat may lead to higher incidences or risk for heart disease. Very different result from a very small change in the chemical itself. Pretty interesting. All right, now there is something called essential fatty acid. Remember what I said about essential in the beginning? Essential means something we cannot synthesize, but yet our bodies require them in order to survive. Okay, so this is a form of lipids that is not for storage of energy per se, but we need it sometimes for other purposes. Enhancing chemical reactions in the body, etc., or not as hormones. Not talking about that. For example, omega-3. This example right here is essential fatty acid. Okay, it's not a storage of energy, but it helps with a lot of different reactions in the body that we're not going to go into. Okay, so like I said, essential means in biology means that we need something. Okay, we don't synthesize it. So we have, for example, essential amino acids where we cannot synthesize those particular amino acids in the body, and we have to ingest it from another organism. Okay, so either we can acquire it from the environment or acquire that particular thing from other organisms, and in this case we can acquire it from, for example, salmons, etc. Okay, so this particular example of essential fatty acid is what we commonly known as omega-3. These are good examples of polyunsaturated fat. Okay, you see how there are more than one carbon double bond carbon. Okay, so that's what we call this polyunsaturated fat. Okay, because you can see that there are multiple number of double bonds within the molecule itself. And polyunsaturated fat is really good for us because of their bulky, unique three-dimensional shape. Okay, they can help to reduce the risk of heart attacks, lower triglycerides in blood, decrease blood pressure, and prevent abnormal clot formation. Okay. So notice that we didn't put energy storage there. Okay, they are for somehow help with the body in staying healthy this way. Okay, with all these polyunsaturated fats. All right, so moving on to the second subtype of lipids called phospholipids. So these lipids are not for energy storage. Okay, they are major constituents of what we call the plasma membrane. For example, those that surround or form a border around the cell. Usually we see the cell as something like this, okay, with the nucleus in the middle, and there's all kinds of organelles around the cell. And so this border right here and the border that surrounds the nucleus is what's formed by phospholipids. Okay, and we'll talk about what these are. Okay. So each phospholipid molecule is made up of a glycerol molecule. So this part of the molecule is the glycerol, as we have seen in fats. Okay, so that's still made of one of them being glycerol, but the glycerol is actually attached to another group called the phosphate group. You should recognize this particular PO4 molecule because it is another functional group that is abundant in nucleic acid as well. Okay, so phosphate group along with the glycerol group come together, and we call that to be considered as the hydrophilic head of the macromolecule. Okay, hydrophilic head. When what we mean when we talk about the difference between hydrophilic and hydrophobic. Hydrophilic means it tends to like water, whereas hydrophobic, it doesn't like water. So phosphate group and glycerol is what makes this particular part of the molecule likes water. That's why it's called a hydrophilic head. And then at the bottom of this molecule, we have two fatty acid. One is a saturated fat, okay, fatty acid. That's why it's linear long chain. The other one is an unsaturated fatty acid. Unsaturated, that's why there is this kink right there, a bend in the fatty acid molecule. Now, this is an important aspect of phospholipid that I will talk about in a bit, but because this part of the molecule is made up of lipids, which are hydrophobic, like we have just mentioned, this section of the molecule is called the hydrophobic tails. Okay, so a phospholipid molecule is made up of a hydrophilic head that comprises of your glycerol and the phosphate group, and the other part of the phospholipid is called a hydrophobic tail, which consists of one saturated fatty acid and one unsaturated fatty acid. Okay, you must know these different components of the different macromolecules. Now, because part of the molecule is made up of lipids, which are hydrophobic, okay, you have this section of the molecule not liking water and this part of the molecule liking water. This is a very important concept that allows phospholipids to be able to form a border around the cell, okay? Now, so because you have a hydrophilic head and a hydrophobic tail, one single molecule but two different type of interaction with water, we call it an amphipathic molecule. Amphi usually meaning both, both of something. So that's why amphibians, for example, are talking about organisms that live in water and land. They live in both environments. So amphipathic mean they're react in two different situations with water. One part likes water, one part doesn't like water. Okay, so amphipathic means it has two different type of reaction with water within the same molecule of phospholipid. Now, the image here shows how these phospholipids come together to form your plasma membrane. So this is sort of like a zoom in image of, for example, if this is the cell, okay, with the nucleus in the center. Okay, you saw those in your lab when you looked at microscope lab when you looked at cheek cells. Okay, so there are organelles within there. So we zoom in, okay, to this particular part, the plasma membrane or the border around the cell, we will see two layers like this. Okay, we will talk about the plasma membrane in detail in chapter 5, but the plasma membrane is made up of two layers of phospholipids, one layer here and one layer here, that come together like this with hydrophobic tails facing each other inside of the membrane, whereas hydrophilic heads right here face either the outside of the cell, okay, outside of the cell, or hydrophilic heads facing inside of the cell. If you think about it, usually outside the cell is fluids, okay? Our body is made up of a lot of water and fluid. So in order for our cells to be able to, live in that kind of environment, you have to have the hydrophilic side facing the outside And then the inside of the cell is usually made up of fluid as well, called the cytosol. So more like a more viscous gel-like formation inside the cell, but it still has water in it. So that's why inside the cell would also have to be faced by that hydrophilic side of the phospholipid, and then the hydrophobic end of the phospholipid will face inwards toward the middle of the lipid bilayer. Okay, usually we call the cell plasma membrane a phospholipid bilayer, is because we have two layers of phospholipids that come together. Many of these one amphipathic molecule make up the entire surface of the plasma membrane, three-dimensionally speaking, around the cell. Okay. And, this is an important concept in terms of where the hydrophobic tails are and where the hydrophilic heads are. Will become an important concept when we talk about why our cell's plasma membrane allows some things into the cell while blocking other things from entering the cell. And our plasma membrane actually does not look bare like this. Okay, it's not empty. A lot of times, okay, it's not all just filled with phospholipids all over the place, but they are actually interrupted by many, many different things within the plasma membrane itself. Okay, there are numerous amount of molecules that are sticking into the plasma membrane or sticking out of the plasma membrane, going through it or going partially through it, etc. You see this big thing going through the plasma membrane. We'll learn about all those things that that goes through, that makes up the plasma membrane other than your phospholipids, when we get to chapter 5 on plasma membrane. Okay. All right, so it's been an hour, so let's call it a break here, and we will end part 1, and then we will continue with chapter 3 in part 2 of the lecture video.